Dear learners, welcome back to NPTEL online certification course on novel drug delivery systems. I am Dr. Satish Devanapelli, Assistant Professor at Department of Pharmaceutical Sciences and Technology, Institute of Chemical Technology, Mumbai. In our novel drug delivery systems, we started with the topic of ocular drug delivery systems. In that, in our previous sessions, we have learned about what are the ocular drug delivery systems, and then we have seen the importance and application of the various drug delivery systems, and then we have seen the what are the conventional ocular drug delivery dosage forms, and after that we have discussed the two sessions on the novel ocular drug delivery systems. In the first session, we have seen what are the in-situ gels and what are the drug coated contact lenses, and then we have seen the inserts. And in our recent lecture, we started with the various other types of the novel ocular drug delivery systems. In that, we have discussed on the particulate systems. In that, we have discussed on the nanoparticles and other various types of the nano based drug delivery systems. Also, we have discussed on microneedles and nano vapors, and then we have discussed on the iontophoresis. Today, we are going to discuss the evaluation of ocular drug delivery systems. As we know that these characterization or the evaluation or quality control studies of any formulation can be classified into three categories. So, the first category is in vitro characterization, and the second is ex-vivo, and third is the in-vivo studies. So, in the in vitro study, we do lot of these studies based on the glasses or by using enzymes. We do all these studies like at artificial conditions, which are called as in vitro studies, whereas ex-vivo we take some biological membranes or some of the tissues and we perform in the physiological conditions, which are called as ex-vivo studies. Like for example, the France diffusion, by performing the diffusion of any formulation by using biological membrane. So, that is called as ex-vivo, whereas in vitro we perform various types of studies. For example, we perform the dissolution study is one of the in vitro study, whereas in vivo we try to see the safety efficacy of the formulation on the animals, or sometimes which may also called as pharmacokinetic, and when you see the efficacy, which is called as the pharmacodynamic studies. So, today we are going to focus on the evaluation of ocular drug delivery systems, which is classified into three categories: in vitro, ex-vivo, and then in vivo examinations. So, here first class is in the in vitro examination or in vitro analysis. The first foremost point that we need to check the sterility, and after that we have to see the quality of the final products. So, these are the various in vitro tests which we need to perform before releasing the products into the market. So, but it depends on the type of dosage forms, and also it definitely depends on what type of dosage form you are converting, whether it is solution or you are making a gels or some semi solids or some type of injections. So, entirely the story of the examinations will depend on the dosage form. So, we will see one by one. So, the first test is nothing but sterility test, and we know that sterility is nothing but free from all viable microorganism, that is called as sterility. Now, how we will get a sterility? So, when we call sterile products, After performing of sterilization by using of heat method, autoclave method, or by using of dry heat method, and by using of various types of the sterilization techniques, which includes UV radiation, gamma radiation, and also membrane filtration, and various types of chemicals we use, and we do the treatment, and then you can kill the microorganisms, which is nothing but sterilization. After performing of sterilization, we can say that our product is sterile. So, still we need to see the quality of our pharmaceutical products, especially the sterility test is preferably important for two types of the dosage forms. First is the parenteral and second is the ocular. So, then which formulation is called as sterile pharmaceuticals. So, here we are going to see the absence of microorganism in the finished product. So, this can be achieved by controlling the bioburden, nothing but the type and the number of microorganisms which present in your product, which is called as bioburden, and that need to be performed under the aseptic conditions. For example, which is also known as the clean rooms. So, here the particulate matter as well as the airborne microorganisms are controlled. So, aseptic room is nothing but, or clean rooms are, which the airborne particles as well as the microorganisms need to be controlled in that environment. Then only we will call as clean rooms. This is further classified into class rooms, also clean rooms 100, 1000, 10,000 and lakh. When number goes, the number of the particles and the microorganism restriction go down. So, clean rooms have, we can say, the which are the clean room 100 are the aseptic rooms. So, here we will develop the sterile pharmaceuticals. So, here what we do, after the developing of pharmaceutical dosage forms, we need to prove the sterility of the any type of sterile product, including parenterals as well as the ophthalmic product. So, here we know this is well known microbiological experiment. Here we perform the growth promotion test. So, here we will see the growth of microorganisms by providing the various cell culture mediums. So, that will help to growth of the microorganisms. So, here we use same principle and same methodology, and we will see the presence of microorganisms in our formulation. If it gives any type of positivity in the growth of microorganism, nothing but your product is non-sterile. If there is no evidence of microbial growth, which is your product is sterile. So, by simple streaking method, we can understand the presence of microorganism. So, here the principle, here we provide the favorable conditions, that means required medium, and also we provide maintain humidity, and also we provide temperature, and that culture medium will help to growth of the microorganisms if microorganisms are present in your formulation. Here we use two types of culture medias. One is fluid thioglycolate medium. This is called as FTM. Some cases it is also preferred as a FTGM. Now, here these we will use for the anaerobic bacteria as well as some type of the aerobic bacteria. And the second medium we use soybean casein digestive medium, SCDM. So, this is we use for the both fungi as well as anaerobic bacteria. So, each formulation need to be performed in two different culture mediums, right. So, here what we do in a general principle, what we do, we take our formulation and here is the culture media, and yeah. So, here this is the container and this is your culture media, and this is a sample. You need to take it and you need to mix it in the container, and we have to keep it at different conditions, that we need to provide the temperature and we need to incubate for 14 days. After that, if you see any growth in your culture media, that means your product is not sterile, or you can say non-sterile. So, if there is no growth of microorganisms which is not there in your formulation, that means your product is sterile. So, we have to remember this general principle. Now, we will see further these tests can further classified into the two categories. One is membrane filtration method, second is direct inoculation method. So, in the sterility test we have two experiments, one is membrane filtration, second one is direct inoculation. So, by seeing this you will understand here we need to use filter membrane and that sample need to be filtered, then that will further pass into the cell culture medium. So, here we use the membrane which has a pore size of 0.45 micrometer. So, after passing your sample, now you can see in the pictorial representation. So, this is your sample, you need to filter. So, the whatever microorganisms which are there in your formulation, if it is a non-sterile, then your microorganism will retain on that membrane. Later that membrane need to be cut into the two pieces. One piece will go to the FTM medium, fluid thioglycolate medium, and the another piece will go into the soybean casein digestive medium, and we need to keep it for 14 days incubation. And here we can see that no turbidity, no sample contamination, right. So, that is in both cases you do not see any contamination, that means your sample is sterile. And in direct inoculation, here also what we do, we simply take formulation and will directly inoculate with the medium. So, here there is no filtration and there is no dilutions. We will take formulation, our formulation, and we will incubate or inoculate with the culture medium directly, you can see in the test tubes. So, here we mix the sample and then we will keep it for 14 days, and if you find any turbidity, that means your product is non-sterile. So, we need to discard the entire batch or we need to repeat once again to recheck the sterility. So, if any turbidity comes, that means your product is non-sterile. So, the second test is pH measurement. As we know that we are discussing from last 4 lectures, the tear fluid which has the pH is 7.4. So, to mimic that conditions, we need to always prepare our formulation according to the or near to the 7.4 pH. So, pH of the eye drops is important for drug solubility and for the stability of the some drugs. There are the drugs which are not suitable to solubilize at this pH. So, there are the drugs which are not stable at 7.4. Such cases we have some range, we can play within this range. pH 6.2, 7.8 is preferable, so that your patient should not undergo any compliances. Yes, if you go below the or higher than this pH, if your formulation is acidic or alkaline, so patient may undergo in the tearing, also discomfort and pain in the eye. So, your formulation need to be at this range. So, this can be performed by using the pH meter. Now, second is clarity test. This is speaks about the transparency of your product. So, this can be performed by using the visual observation. This people will do by the experts only, not by the common people, those who are expert in this field, and those people can compare with the standard formulations, and here this cannot be performed for the suspension. This study is for only the solutions, or you can say the eye drops, and this can be performed by using of the visual inspection. So, here we can use background of white and black color. Here you can see the person is checking the clarity of the product by putting the samples on the background of the white and black background. You can see here is the white background and here is the black background. With that we can see the presence of the visible particles, and there are the instruments we will see in the upcoming slides also. We can quantify the presence of the particles in the formulation. So, yes, by measuring the transmittance, by using a UV spectrometer, we can also by calculating that we will understand the presence of the particles or turbid in the formulation. The fourth one, examination of size and morphology of particles. So, here few studies we are already discussed in case of nano formulation. So, the main goal to understand here what is the size of the particles, the shape and the number of the particles is very important, because the pharmacopoeia guidelines suggests that if you have the various types of the ophthalmic formulation, if you have a formulation, if the particle size is greater than 10 micrometer, the limit is 50 particles per ml. So, similarly, this is for the USP, and if the particle size is greater than 25 micrometer and the limit is 5 particles per ml, similar to the particles which are more than 50 micrometer, the limited number is 2 per ml. So, there is no as such recommendation greater than 500 micrometer. This is according to the USP. Now, according to the European Pharmacopoeia, so we can see here what are the requirements. So, that is why we should know what is the size as well as shape and the number of particles present in your formulation. So, by using of optical microscopy, light abstraction test, dynamic light scattering, laser diffraction, electron microscopy, SEM, TEM, AFM, and also with the counter method, also nanoparticle tracking analysis, we can understand the size and morphology of the particles. Now, here we are going to see these are the specifically for the gels only, and especially that is for in situ gelling systems. So, here you can see we need to see the salt to gel transition. This specifically happen only in the surface of the eye because of the temperature and the pH ions which are present in the tear fluid as well as some other mucosal membranes at the eye. So, that helps for the phase transition from solution to gel. Now, you can see here is the one formulation which contains the carbopol as well as chitosan and carboxymethyl cellulose and hydroxypropyl methyl cellulose. By using of this we can make the solution form, and when you after addition it undergo the cross linking. Here is the calcium ions will help for the cross linking of the chitosan solution. So, here these need to be measured by using the simulated tear fluid. So, we need to calculate what is the time, and also we can consider what is the rheology after the transition or the conversion from solution to gel. That need to be performed, and we need to report the data for the further evaluations. And here we have another parameter, that is a swelling index. So, here we use for the various types of inserts, and we know that in our previous novel drug delivery formulations we discussed on the ocular inserts, which are made up of polymers which have ability to absorb the water and slowly release the drug, and it undergo the controlled and sustained release. This property is very important. But to see the what is the swelling index, the time required to swell the formulation. So, how much water it is taking at different time interval, that is also very important for inserts as well as implants. So, this need to be performed, and yes, so this test is performed by weighing the inserts before after immersion into the simulated tear fluid. Now, you can see this is your formulation, and after administration or treating with the simulated tear fluid, that swells, that means it holds the water. So, that is called as swelling index, that need to be performed wherever we are using the polymers, especially inserts as well as implants, this need to be performed. Now, the another one is that is related to the drug, that is chemical properties. So, here we need to identify how much drug is loaded or encapsulated in the formulation. And the second one is we need to know the in vitro drug release, yes, but this is not like a dissolution studies, because we play with the very small quantity and that need to be performed in the simulated pH conditions of the tear fluid. So, in drug encapsulation, after developing of any novel drug delivery system, that need to be dispersed in the required solvents. After that, we need to do the mixing, and further we need to follow centrifugation and take that supernatant and check the quantification of the drug which is loaded and as well as unloaded by using the HPLC or UV or any type of the quantification techniques. So, from that we will understand how much your drug is presented in your formulation. These are very detailed we have discussed in the nano formulations and their evaluation. So, here we will understand how much drug is encapsulated in your formulation. And the second one is in vitro drug release. That formulation, drug loaded formulation, need to be placed in the required buffer or PBS 7.4 a different time interval during the maintaining that mixing as well as temperature, and we need to collect the samples. This may go from 1 hour to 24 hours, sometimes 2 days, 3 days, and it will go for 1 week. If it is an implant, it may go for years also. So, we need to see the release of the drug from the formulations into the your buffer. So, after that take that buffer and do the quantification and perform the required studies, and from that you will understand how much drug is releasing at different time intervals. You can compare your formulation and with your drug. Consider this is your drug and this is your formulation, and if it is a polymers or some of the carriers, definitely it will show the sustained release. So, the last one in the in vitro is stability study. So, we know that we need to understand the storage conditions of the product. So, here the main purpose of the study to provide information change in quality of active ingredient or medicinal product due to the effect of environmental factors, that is temperature, humidity and light. So, this is the common study which is recommended by the ICH, and these are the requirements. At these conditions we will see whether our formulation is stable or not. So, by providing these environmental factors like temperature as well as humidity and in some cases light. So, we need to understand the storage conditions, or with this we can also predict the expiry date of our products at different storage conditions. So, while performing stability study of any ophthalmic or any type of formulations, we can follow these guidelines will help for the stability study. In general what we do, this is the long term study. So, here we can maintain 25 degrees centigrade plus or minus 60% RH or 30 degrees centigrade plus or minus 2 degrees centigrade 65, that need to be performed for 12 months, which is called as long term. Whereas, intermediate we perform a different condition, that is the 6 months, and the accelerated we need to perform this for the 6 months. There are the conditions in the refrigerator as well as freezer. This is also considerable according to the ICH guidelines. So, with this we will understand what is the ultimate the storage or stability conditions of the formulation. If your product is stable, then you can prefer the same condition after the manufacturing. If your product is not stable in the light, we need to provide in the amber color, we need to take required precautions for the storage conditions. So, in the in vitro studies we can perform the stability studies, and here we have one example of ex-vivo permeability assay. So, here what we do, we try to understand the diffusion of our formulation, that is called as ocular diffusion. Everyone we know that. So, here we use a France diffusion cell. So, here we use the corneal membranes. These people also use for the gastric mucosa as well as skin tissues, various types of the even nasal mucosa membranes also we can place, and we will see the how much is drug is moving from this place to and to the your receptor chamber. So, here what we do, so this is the France diffusion cells. So, here is the receptor and this is the donor chamber, and we will keep this assembly at a temperature of 37 degrees centigrade, and we also provide some the stirring also, and after that we will place this cornea which is sacrificed from the animal eye. So, after that we will place this and also we keep the donor here, and then we will place our formulation into this. At different time interval we will collect the sample and also maintain the sink conditions of the formulation. So, after that we will see the how much is drug is permeating from your formulation. So, here we need to take the eye and from that sacrifice it and take the cornea and then do the diffusion studies. So, we can take cornea as well as sclera. So, whatever the drug is permeated you will see here, and that need to be quantified by using UV as well as HPLC method. So, here we have 3 in vivo examination. We will spend 3 to 5 minutes and we will try to wind up the evaluation of the ocular formulations. So, yes, this is one of the most preferable method, eye irritation test. We do it for the even for the various types of the topical and transdermal patches. This is the skin irritation, or you can say modified Drey's eye test, and the transcorneal permeation study and also pharmacokinetic and pharmacodynamic studies are there in the in vivo examination. So, here simple experiment, we will take the albino rabbits that need to be examined and weighed, and we need to take small amount of formulations and that need to be applied on the eye, preferably right eye, that is a test sample, and the whereas other left eye you can keep it as a control, you can give some saline. So, we need to close the eyelids gently, and then after that we need to see the observation of the eye. So, whether it is blinking or is there any rubbing and also any irritation, and that we need to consider. The observation is that, so we need to examine the rabbits under the proper light with the magnifying glasses and the slit lamp, and we need to give fluorescein dye as well as photography. So, that need to be used, and with this we have to visualize the eye. If there any discomfort, any problem in the blinking of the eye and rubbing, that need to be counted at different time intervals, and based on that we can consider whether it is the irritative or not. Now, you can see this is a simple example people have studied for one product, marketed product, as well as they have prepared the liquid formulation. The reference is given below. Now, you can see they have compared the control as well as their 2 formulation and the marketed product at 1 hour, 24 hours, 72 hours and 14 days. You will see there is no problem at all, there is no change in the physiology of the eye. So, whatever you are seeing the red color, that is the dye. And yes, in vivo transcorneal permeation study. So, here what we do, we put our formulation into the conjunctival sac, and after that we will need to anesthetize the rabbit, and we need to extract the aqueous humor, and further that need to be treated with the zinc sulphate. So that will precipitate the protein which are presented in the aqueous humor. Then we need to centrifuge and quantify the remaining amount which is presented in your formulation. So, here in vivo pharmacokinetic and pharmacodynamic studies, we perform pharmacokinetics to see the drug absorption, distribution, metabolism ADME, and in the pharmacodynamic we will see the effect of the drug on the body or the at eye site. So, with this we can predict the efficacy, safety as well as dosing regimen of the formulations. The overall in vivo pharmacokinetics and pharmacodynamic studies we perform for various ophthalmic products to see how your formulation is responding to the disease and as well as how your eye or body is responding to the formulation or the drug loaded formulation or to the drugs. So, these are the various types of the evaluation studies. So far we discussed on the in vitro evaluation studies, ex-vivo evaluation studies, and then we have seen the in-vivo evaluation studies. So, with this we are concluding that we have completed the entire portion of the ocular drug delivery systems. So, in our upcoming classes we are going to see the ocular drug delivery systems. In our upcoming classes we are going to discuss on various novel drug delivery systems. Thank you so much.